For the participants who have just joined, please feel free to introduce yourself in the chat box with your educational background and research interests. We will be starting in 2 more minutes. All right, so let's start with today's webinar. Thank you for your responses, for introducing yourself in the chat box. All right, so hello everyone, a very warm greeting to one and all. My name is Sri Gauri Krishnakumar, and I would like to welcome you all for today's webinar on data science for biomedical research. So why do you need data science for biomedical research? Biomedical data science involves the analysis of large-scale biomedical data sets to understand how living systems function. The rapid growth of high-throughput data, including omics technologies, gave rise to significant demand for data science skills and experience along with bioinformatic methods of analysis. That is why we have designed this online training program that will cover practical and conceptual aspects of data science, including data wrangling, statistical analysis, and machine learning in application to high-throughput biomedical omics data using big data analysis tools such as the T-BioInfo Platform, R, and Python coding languages. Before proceeding further, we also have an exciting announcement about the upcoming hackathon on microbiome in health and wellness. This is an initiative to provide students with a platform to showcase their innovative mindset of problem solving. Here, each student will have the opportunity to develop a mini project around the hackathon topic, form a team with at least 2 other students, and compete against each other for mentorship, support from established companies, and long-term interest from the business community. The applications of hackathon will be in the field of obesity and precision nutrition, innovation and beauty products, and pregnancy. So I would like to urge all the participants to be a part of this hackathon and make the most out of such a wonderful opportunity. If you wish to know more about the event details, you can visit the link that will be provided to you by one of our team members. So before we begin with today's webinar, let me also take a brief moment to introduce you to the team behind the program. We are a US-based bioinformatics company who is working with multiple academic and commercial collaborators to develop easy-to-use analytical tools, and our mission is to make bioinformatics more accessible. The OmicsLogic training has been completed by over 24,000 participants from 187 countries in over 300 workshops. And due to this fast growth, our team is working with local and regional coordinators that are helping refine local program logistics and adapting them to the needs of students and researchers around the world. This training has been completed by participants in 6 different specialization tracks, which includes oncology, infectious diseases, precision medicine, neuroscience, data science for biomedical data, and comprehensive training on omics data analysis. So how do OmicsLogic resources help advance bioinformatics teaching? OmicsLogic is a portal for practical and theoretical learning of bioinformatics. It takes a combination of training modules, data analysis tools, curated project datasets, and interactive sessions with mentors to give the student a clear path up the Bloom's taxonomy pyramid. No matter what the user's initial experience with bioinformatics is, OmicsLogic is a useful tool for gaining a baseline knowledge of the theory behind bioinformatics, analysis of large data sets, and introduction to basic coding languages such as R and Python, or even beginning a data analysis project. The program comes with an access to the OmicsLogic courses, which includes courses on bioinformatics, precision medicine, multi-omics, which includes genomics, transcriptomics, metagenomics, and epigenomics, data science that includes bioinformatics using R and Python, as well as cheminformatics, and example projects of different fields which you could learn from and replicate for your research work. Users will be given directions to access the T-BioInfo Platform for bioinformatics processing and analysis of data. The platform includes demo pipelines as well as data management and analysis cloud infrastructure to run bioinformatics pipelines. Different stages of the analysis are performed in different sections on this multi-omics platform. So first of all, let's begin by signing up on the OmicsLogic Learn portal. Before that, let me walk you through a brief demo on what a completed profile on the portal looks like. So once you sign up on the portal, these are the features you need to update on your account. That is, a completed profile will have a profile image, full name with appropriate capitalization, link to your social media handles, which could be your Facebook, LinkedIn, Instagram, OrcID, or even Twitter, and a brief bio about yourself that will consist of your educational background and research interest. The mentors can view your progress under the Activity tab. The courses you are currently completing will appear under the Courses tab. And finally, after the completion of your course, the certificates will appear under the Certificates tab. So with that, let us begin by signing up on the portal. I'm sharing the link in the chat box. Please click on the link and I'll walk you through the process behind signing up on the OmicsLogic Learn portal. So here is the link. For users who do not have an existing account on the portal, please click on the Create an Account option that you see here. So here you will be asked to enter your name, email ID, and provide a password. You also have the option to sign up using any of your social media accounts, which could be your Google account, Facebook account, Apple account, GitHub account, or even your Twitter. And with that, I'll pause for a couple of minutes for the participants to sign up. Once you have signed up, please put a yes in the chat box. And if you're facing any technical issues for signing up on the portal, you can share that in the chat and our team will be happy to help you. So I'm monitoring the chat. Once the participants have signed up, please put a yes. If you're having any queries, you can put that in the chat as well, and I'll be happy to help you. So meanwhile, since I already have an account on the portal, I'm going to click on Login Now option that you see here and click on the Sign In option. All right, one person has signed up. I'm waiting for the rest of the 20 plus participants. So once you sign up on the portal, you're able to see the various courses and example projects that you can browse through after the webinar gets over. So since most of the participants are interested in data science, let us enter the keyword data science and see what are all the courses and example projects that will appear. So as you can see, there are a lot of courses on Python, R course, as well as example projects that you can go through. Now, the most important step that I discussed previously was setting up the profile. So for that, click on the tab that says Welcome Back with your email ID. And here you will be able to see the feature, profile edit icon. Here you'll be able to edit each of the features that you wish to upload on your account. So here you can insert your educational background and your research interest. And in my case, I've linked my LinkedIn account and my OrcID. So here under the Activity tab, the mentor and the community manager of the program will be able to track your learning progress. And once you start completing the courses, it will appear under the Courses tab. And once you complete a project and upload it on the portal, it will appear under the Projects tab. And once you enroll for any of the program, for example, the data science for biomedical research program, it will appear under the Programs tab. And finally, when you complete any of the courses, the certificates will appear under the certificates tab. So with that, if anyone has any queries, please put them in the chat box. Otherwise, we can now move on to the next part of the webinar, which will be taken over by Dr. Raghav, who is the mentor and research consultant at Biomedical. Over to you, sir.
Thank you very much, Sri Gauri. So while you are busy signing up, let me find ways to share my screen and set up for today's presentation. So can you see my screen? And can you hear me clearly?
Yes. Yes, sir. Both are fine.
Okay, thank you very much, Sri Gauri. So let's let me welcome you one more time for today's session. Today is the first session for the data science for biomedical research. And let me set up the chat box so that I can follow you, responding to my queries. Or if you have any questions, please type them as we go along in today's session. So in this program, we will focus on the methods for the analysis of big data and data wrangling and data pre-processing and data analytic skills for bioinformatician, as well as those who are interested in other types of non-biology related research analysis using the data science or data analysis methods that we will be focusing in this program. So in today's session, we are going to discuss about some of the key topics, overview of the key topics, and we will discuss about what is the data that we are going to be analyzing. We are going to discuss about what is the type of the data that we are going to be covering, and we'll touch upon data loading and data preparation slightly, and we will also briefly breeze through some of the statistical tests that we can perform on the data, and we can also discuss upon where to find this data. A couple of slides on where to find this data, and generally in this data science program, what are the omics data types that we are going to use for our analysis and our understanding of the different data analytics methods that we are going to use. So what is data science? Right? Data science can be thought about or can be abstracted as a convergence of data management, data manipulation, and analytical methods for these hypothesis testing in any specialized domain. And this time, for the bioinformatics and for biomedical research, related research, the domain is the biology, and the methods that we are going to use mainly based on statistical analytical methods and informatics tools to manage the data that is going to flow into these methods and that's going to come out of these data analytical types to infer important questions, important biomedical research and biomedical related questions, to understand patterns, to understand the disease biology, and to understand various different questions that one can set up using the data and using the research theme itself, right? And basically, because of these methods, we will also encounter and we will also discuss that the domain biology itself is rapidly acquiring the characteristics of data science. And when I speak about that, biology in today's era contains billions and billions of data points on genes, proteins, and other molecules. And these data points are compiled in large files and are systematically studied to understand the patterns in the data. This type of analyzing the data will actually lead to more knowledge and more understanding about human health, about biotechnology in general, about agriculture, and about livestock, and various other different research areas. So this huge volume of data and its influx actually generated tremendous development in associated methodologies, and these together has completely changed the biological research towards the data-driven research or data-driven approach for making bioinformatics for life science as one of the prominent methods to understand biology as such, right? So this is completely different from what has been traditionally practiced in biology, as biology has traditionally been a hypothesis-based and a data collection-based research domain. So over the past 20 years, as I mentioned, there has been a phenomenal growth in the adoption of high-throughput sequencing technologies and high-throughput technologies like next-generation sequencing. In research, in clinical practice, in biotechnology and biological pharma industry sectors. And this adoption of this technology has actually produced an exponential growth of data that is publicly available. And these data are commonly referred to as omics data. This trend has actually produced a big data problem that preludes many clinicians and many biologists from understanding knowledge and understanding pattern from this data regarding the nature of, for example, malignant tumor and malignant processes and the relationship between clinical phenotypes and their molecular drivers behind them. Yeah. So for example, if you take in today's era or in the last 3 or 4 years, due to the pandemic and due to the availability of the methods to sequence, and we can generally see this phenomenal growth or phenomenal increase in the volume of genomic data or full genome sequence data specific to COVID-19 or coronavirus when you compare it with the other well-known virus that has been already studied until now. So we spoke about omics data. Omics data generally includes phenomics, genomics, epigenomics, and technologies like next generation sequencing and high throughput sequencing can actually be used to explore the roles, the relationships, and the actions of various types of molecules that make up the cells of an organism. Many types of these omics data can be generated using NGS method and what is also commonly referred to as high throughput sequencing methods. And these data can actually show detailed information about patterns of the gene expression and isoform expression, detailed information about genomic variants and its effects in the phenotype or in the disease biology, and general information about, for example, amount or level of gene expression patterns and gene expression counts in relationship to the disease biology in general. So we discussed earlier that bioinformatics and biology actually acquires the properties of data science. Now, to give you a brief overview of data science, we know that data science involves developing methods of recording, storing, and analyzing data to effectively extract meaningful and useful information. The goal of the data science is to gain insights and to gain knowledge from any type of data. This type of data or this data can generally be structured or unstructured or semi-structured. Unstructured data is what you typically have to deal with, and when you are acquiring publicly available data, and this unstructured data has to be prepared for analysis or pre-processed for analysis to make it useful and to make it applicable to a variety of different methods that you will be learning over the course of this program to apply to different omics logics, or not omics, different omics data types. So to give you an example of what is an unstructured data and how to force or how to gain structure from this unstructured data, we can have a couple of illustrations. So for example, if I ask you, what is your educational background? And if I let the answer column completely open and free, we will have a variety of different responses ranging from one word answer to a small passage describing what you are studying and what are your interests and what are your main area of focus. But these responses will be very easy for any human to understand, but for computers and for information processing to make sense out of this data is very challenging. So what do? We force structure onto the data that you are going to collect. How can we force structure? We can tend, or we can change the survey, replace the survey into categories that we can easily analyze, count, and use or subject that to different statistical methods. So when we do this, we are forcing structure into this unstructured data for to enable us data interpretation and data analytics to understand the variety and trend in the data, so that we can make sense of what we intended to start with, right? That, and to visualize data also, this is actually usually done. So this analyzed data and visualized data can easily be shared, easily be interpreted, and easily be understood generally. But if you take the unstructured data, it's difficult to make sense out of the results through computational methods. This example, or this instance, or this illustration would be true for any kind of data, and especially big data. So big data has certain characteristics that we have to keep in mind. It has velocity, variability, and volume. All of these has been we can illustrate in the SARS-CoV-2 availability of genome, the SARS coronavirus genome example that we spoke about a couple of slides ago. So before 3 years ago, or 3 years ago, we hardly had any coronavirus or SARS-CoV genome at the scale, at the velocity, at the speed, and at the variance that we can analyze these information now. So this is typical example of what the available technology and the availability of methods can do to the data that can be generated, and that has been generated, big data that has been generated in an explosive manner. And we spoke about structuring data to understand the processes and to understand the trends in the data. So how to structure omics data? In order to structure omics data, we need to take the raw sequencing data output, that is in the strings of letter A, T, G, and C, and we have to generate a matrix or a data set of combination of a continuous and categorical data that can be analyzed using statistical methods. To do this, we organize a specific set of processing steps in sequences that we often refer to as pipeline, analysis pipeline. And these are specific to each types of omics data. So when we speak about data processing as a procedure to generate data set or a structured data, we can term these analysis methods as specific data analysis methods, like genomics pipeline or genomics data analysis method takes the unstructured genomics data, that is in unstructured sequence data, that is usually stored in FASTQ file format, and then applies structure to this data. And similarly, we can apply structure to sequence data that is intended to measure transcriptomics gene abundance or gene quantification, and intended to measure methylation data for gene regulation, and etc., etc. So to be able to run complex bioinformatics pipelines, you need high performance computing or cloud-based servers without slowing down your own computer, right? So in order to understand, or in order to develop a logical understanding of many methods involving this data preparation, we have a specific set of courses and lessons that will show you how to set up these analysis pipeline in order to generate the structured data from this unstructured data. Many of you who are new to bioinformatics and who are interested in these processes are free to explore this also. So there are commonly used methods to study, for example, DNA variation, and sequence pipelines to study, understand genomic variation, gene expression, protein abundance, and compositional properties of microbial communities. All of these are different types of data that we will learn to explore, and we will learn to mine for patterns to understand the trends in the data related to the biological condition or disease biology or phenotypical characteristics of the sample that we started with. So we will also look at full overview of the data science methods to understand how different stages of analysis work together to help define the hypothesis, to help prepare the data for meaningful applications of statistical methods like machine learning and practical challenges. So in this flowchart, for example, you would assume that most of the time in data science methods are spent in the data analysis section. But in fact, you will learn that most of the time and most of the analysis and most of the effort will be dedicated to this section, where we have to process the data and explore the data in order to set up the perfect analysis method that will help us understand the patterns to answer the hypothesis or to answer the question that we have set up at the start of the experimental design. So you will also learn different methods to process the data, different methods to visualize the data for exploratory data analysis, and different methods to analyze the data that suits your research question, that suits your research hypothesis. So we spoke about structuring the data. So let's go through a couple of examples of how to structure different types of data, like genomics data, transcriptomics data, and metagenomics data. And what will you get at the end of these analysis methods, and what is the structured data that you will get your hands on, that you can apply a variety of different statistical methods that include machine learning methods that you will be frequently applying in this program. So to achieve some of the results, we are going to take, for example, in genomics data analysis, we will take the FASTQ files with short reads and run a pipeline, an example pipeline called variant calling pipeline, to get an output of tables, or to get a variant called a VCF file that lists all the genomic variants that is present in the sample that you have sequenced, compared to the reference sequence that we have generally available at the repositories or the libraries. So this processes involves mapping and identifying somatic variant calling steps to produce a table that we can analyze, or a list of these variants. For example, when this shotgun sequencing methods or pipeline is used, we will generate a lot of short reads that are to be mapped onto these specific sections of the reference sequence that you can see here. Since we have zoomed out so far, we cannot read the reference sequence, but these horizontal bars are the stretches of sequences in your sample that are short reads. So when you go through the genomics data analysis, you can understand what we are actually talking to. So, but at the end of this analysis methods, the aim is to identify variances in your reads or in your sample compared to the reference sequence, or compared to the reference that we are comparing with. So at the end, for example, in can you hear me now? Yeah. So in the end, for example, in oncology, we will categorize the non-matches and we will categorize matches for their mutational status, and are often we can categorize them into somatic mutations or germline mutations to understand how important and how they affect some of the genes involved with tumor growth and tumor disease progression in general. So to analyze such a data, we will need to prepare a table of somatic scores or mutational index for each genomic position. This is a table or structured table that one can generally obtain at the end of the genomics data analysis, that you can subject to a variety of different statistical analysis and machine learning analysis to understand the importance of this genomic importance of this mutational index, or importance of this mutational frequency at each and every genomic position. Okay, it is now responding. So another example of such data, omics data, is transcriptomics data, which represents the mRNA expression in a to analyze what are the process going on inside the cell by observing active transcription of protein coding genes, right? The DNA first is actually transcribed into RNA, and RNA is translated into protein inside the cell, as we generally know, and we have we have our exposure to different protein synthesis process inside the cell. So this process has an innate variability that is associated with producing alternative splicing or producing different exon skipping that will generate isoforms and genes, variants of the genes that one can easily associate with the disease biology or easily associated with the phenotype of the samples, and etc., etc. It is very important to quantify the level of expression of these alternatively spliced variants of a single gene and these different genes in specific, in order to understand their effect and their importance and their mechanism that drives, for example, tumor growth, tumor progression, and disease biology, etc. So a typical analysis for the pipeline for RNA-seq data or transcriptomics data analysis goes like this, where you will first pre-process the data by cleaning out some of the adapter sequences, followed by mapping the reads onto the reference genome to understand how genes are expressed and what variations in alternative splicing can lead to transcripts that will define a phenotype. And then in the end, you will quantify the reads that are mapped onto specific genes and their isoforms. So this results in a table of continuous numerical data that is annotated by gene ID along the rows and sample ID along the columns for any given experiment. And at their intersection, you will get gene expression level that is specific to that particular gene ID and this particular sample name. So this is another example of a structured data that you will take in order to apply to different statistical methods to understand the pattern. For example, you can use data mining approaches to study the segregation and to study the sample order, and you can apply, for example, machine learning analysis like supervised machine learning to classify the samples into predefined classes, and etc., etc. Another type of data that I told we will be discussing is the data that comes from same sequencing technique, that is high throughput sequencing technique, but offers a completely different view of the biological sample that we are considering. In this case, we are not looking at one specific sample or one specific organism. We are looking at a collection, or we are looking at a community of millions of microorganisms that is represented by genomes that are very different from each other. In order to structure this data and to organize this genomic information into categories, we can take functionally related gene that is common to all of the organisms, like a ribosome gene, which is compared to a database of known sequences to, for example, identify the identity of these microbes that are present in the sample that we have collected. So this can be applied, or this analysis is commonly referred to as 16S rRNA amplicon data analysis, and can be performed like other analysis methods in the T-BioInfo platform using DATA2 or KIME2 pipeline that we usually refer to. So as a result of this analysis, you can obtain proportions of taxonomic groups across samples with the analysis of sample diversity, or with the analysis of disease biology or different phenotypes in general. So if you start with understanding metagenomics or microbial population differences between two different phenotypes of the sample, then we will find ways to relate and understand the pattern related to these different phenotypes. If you want to understand the disease biology, then we will take metagenomics or microbial community sample from people who are infected with the disease and people who are free from the disease, and this becomes a comparison in general. So yeah. So this data analysis again results in basic type of structured data that is resulting from this type of analysis is referred to as OTU table, that is operational taxonomic unit table across the sample with proportional abundance in each and every cell. So instead of gene IDs that we spoke about in the transcriptomics data, you will have operational taxonomic unit or OTUs across the rows and samples across the columns, and at their intersection you will find abundance of that specific taxonomic unit, which is a species, that is a group of species, the related species at the level of genomic identity percentage defined by us while analyzing. So another information that one can obtain from this analysis is the identity of these OTUs in general. The OTUs are linked to their abundance levels in the previous table, and their identity is actually calculated from the by comparing the sequence, that is sequenced with the database of sequences of all the organisms and their phylum, taxonomic groups of kingdom, like kingdom, phylum, class, order, family, gene, species are actually identified and tabulated. This, together with the previous structured data, we will take and we will analyze and look for patterns of abundance level across different family, order, or phylum at the different rank that we want to understand their patterns in the biology of our samples. So how would you go about doing this? So we discussed along the way, one of the key steps in performing these data science or data analytics methods of omics data is to generate a structured data. Once you have this structured data, we can actually load up this structured data into an environment like R or Python in order to understand their pattern and to subject to a variety of different statistical methods and to make different visualization options from the data that we have in general. So this can be loaded into a two-dimensional table, and that table cannot be a matrix because over the course of last few slides, I showed you that this data may contain columns that are full of numbers, columns that has characters or that has identity in terms of gene IDs, and columns that has OTU identity. So we have a collection, we have a table that has a collection of different data types. Usually such an information is loaded into an object called the data frame in R or Python, which we will explore in much detail. So any data that has a collection of more than one type of data, right? And that can be loaded into a data frame object. And while when you have a data that has specific data type associated with that, you have to use something like a vector or a matrix or array, depending upon the dimension of the data, in order to load them and to process and analyze them. While we also will encounter some other data types like objects like lists, which contains a multitude of different data types at different dimensions. Generally, we will not use list until and unless we are going to perform specific data analysis. Many a time, we will load the structured data into a data frame object and then process this data frame to represent a matrix or to represent a vector that can be applied to different statistical and visualization tools and analysis methods. So this is called preparing the data. Yes, this is called preparing the data. Yes, Srigauri, if you have some questions from the participants, please put them up in the chat box. I cannot read them for some reasons.
Yes. Okay, sure, sir. The first query is, do you offer courses in BioPython?
The Python module that we have will also include BioPython, right? We do have a couple of lessons that involve BioPython in loading sequences and to analyze these sequences and their identity and similarity levels. But we will use a variety of different Python tools like scikit and scikit methods for general data analysis methods, right? BioPython is used mainly when you have a lot of sequence related information, like you can use BioPython for VCF calling, variant calling, file reading and their analysis. But we are going to deal with a lot of downstream analysis after you have generated the structured table. So that might not have a lot of sequence data, but using BioPython related analysis is just one extension of what you can learn here. So we will be dealing with a lot of machine learning analysis that can be performed on the omics data and the structured data that you will generate from here. So BioPython is just one phase or one module that can be used to analyze such data. And do you also have options for proteomics course using AI? We do not have options for proteomics course using AI at the moment, Peter. Communicate Festus, if I did not butcher your name. So, but we do have other omics data type, and we do have methods to integrate these different data types that will also include proteomics data types, right? At the end of proteomics data, or at the end of proteomics data analysis also, if you are going to get a structured table, then that structured table can be used, and that structured table can be applied to different variety of different machine learning data science methods that we will be seeing and that we will be performing in this program so far. So any other question? Yes, Sri has another question. I am analyzing microbiome data using data2, but I got stuck at a stage where I have to perform phyloseq object. Can you, this is very specific question. Can you check the code for me? Please get in touch with our support group at support@pine.bio, and then we will discuss what we can offer to you. Okay, I did not get the name of the person who has this query. And any other questions, Sri Gauri?
No, sir, that will be all. Thank you, sir.
Okay, so let's move forward. So, for example, we spoke about preparing the data, which is an important process in process after structuring the data and applying to different statistical methods. So we will learn how to prepare data, and we will learn how to use different plotting tools like histograms, scatter plots, bar plots to find relationships in the data and explore ways to characterize millions of data points per sample, right? Why do we need to do this? We have to do this in order to understand the pattern, so that once you have an idea of what is the pattern that you are observing in the data, then you can set up specific analysis methods to answer the question that you want to choose. For example, if I see a generalized pattern that I have 3 separate clusters of samples that in my data, or my data segregates the samples into 3 separate clusters, then I can spend time and more effort in characterizing these specific clusters by applying classification tools or applying unsupervised clustering methods, unsupervised machine learning methods to understand what is specific to each of these clusters that I can use for further biological interpretation of these clusters and naming of these clusters of samples. This is fairly commonly used in analysis of any omics data type related to tumor samples, where the effort is mainly to understand the multitude or understand the variety of different tumor types that one can associate with any specific tumor samples. And omics data type is one way to observe their segregation into these different histological types, or these different molecular subtypes of tumor, or any other pattern that can differentiate the tumor data, that can differentiate the omics data of this tumor based on their segregation. This pattern can also include, for example, clinical data like demographic data, like the age differences in the patients. It can also include the treatment responses. So these are all some of the examples and some of the instances which you will be analyzing in the program as we go along. So to understand this, we will use, for example, to understand this, we will use visual representation to spot, for example, technical noise that is important to be eliminated using various normalization techniques and data transformation steps. And the choice of these normalization and data transformation steps will vary and has to change depending upon the data that you have chosen and the data that you are analyzing and the pattern that you will get. We will also speak about making machine learning models more transparent and explainable whenever we present data to our collaborators and to share with other peers, right? This is extremely important in the era where more and more research and efforts are spent towards translational approach of any data analysis. So we have to make the models more explainable in terms of basic data that has been collected. If you are collecting or if you are analyzing transcriptomics data, the machine learning model that we have built should give you or should result you in a set of important genes that are very, very important, that are crucial to perform this accurate classification. If you are, for example, analyzing genomic variance data, the machine learning model or unsupervised clustering that you have built should actually explain which mutations are prominent and are very important in classifying these samples into these different clusters that we have identified. So we will also have specific chance, and we will also have a lot of opportunity to see how different data mining methods and machine learning methods are used to group data objects by, for example, feature similarity or similar expression patterns of genes. And we will spend a lot of methods to train a model for predictive analysis. And we will see what are the different data mining approaches like hierarchical clustering, k-means clustering, and how they differ in analyzing your data, and what is the main underlying analysis method that differentiates these two different data mining approaches. And we will also discuss a variety of different classification machine learning tools, like ranging from as simple as decision trees to as complex as random forest methods, while discussing SVM, LDA, stepwise LDA, increasing the order of difficulty in the complexity to understand how these different methods and when and where to use these different methods depending upon the data. So it depends upon the volume of the data, it depends upon the variety of data as we spoke about, and depending upon these characteristics of the data, you have to choose appropriate data mining and classification tools for the model and for the analysis that you are building. And all of this we will discuss with specific project examples and publication examples on curated data sets and publicly available data sets also, so that you can get a good handle on the methods that are used and the questions that it is related to and the type of analysis that is being developed for this answering the question. So how do we do that? At the basic or at the heart of all this is to load the data and visualize this data using relationships and spotting relationships and patterns in the complex data. So we will go through some of the or at least one specific code lesson to give you an idea how this lesson in R is actually structured. So let me reshare my screen. So how to do that with stop share. Okay, that is my Zoom. Srigauri, can you see my screen? Can everybody see my screen?
Yes, sir. We are seeing the program page.
Yes, that is what is intended. So yes, this is a program page where we have all the other associated resources. So for the resources that we are discussing today, we will go about data processing and visualization, and yeah, so you can check some of the associated resources in this visualization. Yeah, this is the lesson 1, loading data in R. So this lesson is actually structured. This lesson is actually structured as section to understand the code, learn the code, and where each and every piece of code is actually properly commented and properly annotated to tell you what different parts of this code do and how we can, for example, load data onto into the platform like R and Python. And we have sections where you can try the code that you have learned so far. So this actually runs on a R console that is loaded when you load this program page, when you load these lessons in the backend. So for example, in this practice code, what have we done is we have actually erased, or we have actually made deliberate mistakes in the syntaxes and in the commands that you will need to run these code successfully. So if you run this specific code as it is, it's going to throw you an error. And most of the time, this error is not going to be meaningful as it is with many programming language. So it's important to read through what is the intended challenge to practice in this specific area. So it says you have to copy the file link here, copy link address, and then paste it here so that we can refer to the input data that is going to be loaded. So I paste it. So that now the input data is specified. Let's clear the console output and then run it again. Again, it gives me an error. This is because we have made some of missing parts, or we have deliberately deleted some portion of the syntax, so that you can spot it and then make necessary changes as you have learned from the learn the code section. So it goes to read.table, and again I clear this and then run this. So now you get success in running the code and the answer is correct. Many a times you will also see that sometimes you will get responses like success in running the code, but answer is incorrect. That would mean that there is no problem in the syntax, but there could be specific answer that we are expecting you to produce from these code blocks, that you will have to check your codes to produce to exactly address the specific part of challenge that we want you to go with. So that is what I wanted to show you today in terms of how this is actually organized. And let me reshare my screen. What happens? Okay, can you see my screen?
No, sir, we can't see your screen.
Because I also cannot see this. Oh, I think I got it. I was sharing a different portion. Yes, now can you see my screen? I think I got confirmation, and you can also see my screen.
Yes, sir, we can now see.
Okay, perfect. So this is a snapshot or a screenshot of what you can do with the coding lessons, and this is the same organization for both R and Python courses. So for exactly what you saw in R is replicated in Python, where you will learn the code and you will address necessary we will address necessary parts of these codes, and you will have separate section where you will load Python console and then fulfill or actually perform or work on these challenges to complete the challenges successfully. So what do we want to do? How do we want to do this? So in order to understand the application of all we have discussed so far, we will be able to share with you a couple of specific research projects, and that is reference or that we take from publicly available resources, and we get from publications that you will be redirected to, right? For example, in this publication, I'm not going to go over with a lot of details. If you read through this, you will get information about what are the methods that are being used, what are the data types that are being used. So we will take this publication and take not all the data, but a curated list of data, and perform some of the data analysis methods in order to verify their findings, or in order to reach at a different finding that we have set up or we have started to analyze this data. So for example, in this specific publication, they have used a lot of omics data types, like 7 different molecular profiling omics data types, and then associate that with the treatment response data in terms of IC50 values, and then perform multi-omics data integration in order to study treatment responses for several of the patients that they have analyzed. So we will take this data and then discuss by building up the level of complexities and increasing the integration of different omics data type in order to first understand the patterns of the omics data or structured data at its simplest level. We take one data type, study the pattern, and then we apply a more sophisticated analytics method and study how the patterns are evolving, and then we can build up on this method by including some more another molecular omics data type, and then finally we will merge 2 or 3 different multi-omics data types to understand their pattern in relationship to treatment response, for example, in this specific project. So this and many other information is available over right throughout the program page, and you are free to check out the program page and and what the program has to offer. So with that, I conclude, and I actually give the session podium back to Sri Gauri or Sonalika, who is going to take over.
So we do have few more queries. That is, Kadambari is asking, what are your suggestions for beginners about how to understand the big data? I am not from IT background, just starting with studying C++.
Okay, Kadambari, I think you have to start with courses like Introduction to Bioinformatics and Bytes and Molecules. So what these two courses are defined, or what these two courses are designed to do, is to introduce you to generally what big data bioinformatics is, and what are the different domains and research fields that they are applied to, and what are the, for example, translational end product that you are going to get by applying these methods and these to these data to different research fields. For example, in the health sector, the idea is to improve the diagnosis and improve the prognosis prediction of the deceased patients, and to understand which or to apply that to precision medicine or personalized medicine, where you can identify patients who are going to respond to any treatment that is being considered. So these types of relationship between the data, the method, and the domain and the application is actually given in the Introduction to Bioinformatics course and Bytes and Molecules course. By the end of these two courses, you are expected to get a clearer picture of your interest in terms of domain, in terms of research field, in terms of data analysis methods, and in terms of data in general. Once you have this clarity, then you can probably sign up for this specific data science course, where we will focus on the analytics method that is applied to a variety of different omics data types, as I actually mentioned to you about. Or if your idea, or if your understanding, or if your focus is towards gaining extensive analysis knowledge in analysis methods and different methods to specific data types, then we have programs that addresses that point also. We have specific genomic data analysis program, we have specific transcriptomics program, and we have other programs that you want to check out their announcements and etc. So yeah, I think that partially answers your question. So if you are new to bioinformatics, and again, I have another question in similar line. I will take Adivale's question also. You have knowledge in IT, but no knowledge in biology. You and anybody who wants to get an introduction to bioinformatics should start with introductory courses like Introduction to Bioinformatics and Bytes and Molecules. So you will have plenty of exposure to big data bioinformatics and different methods and different research field, as we mentioned to you about. So if you have knowledge in IT, and if you want to learn how these bioinformatics processes and methods are actually applied to biological data without spending a lot of effort in understanding the underlying biology, I think this data science program can actually suit you very well, because it is going to give you what are the necessary, for example, perspective that you need to address and that you need to focus on in applying the known data science methods that you might have already have knowledge in, to be able to successfully handle omics data types and big data bioinformatics data types. There are some basic underlying data structural differences in omics data types that we will specifically address, and how they actually drive the choice of method, choice of analysis methods, and choice of different analytical methods, and choice of classification algorithms, and unsupervised algorithms also, depending upon the structure of the data. For example, to give you one specific examples, in any Internet of Things or in any financial data, you will have the number of observations to be huge and number of features to be very low. But in the omics data types, in structured omics data type that we have discussed, the number of observations is fairly low, and while the number of features is extraordinarily high. So this proposes a specific challenge that you will need to address while setting up the parameters for very well known unsupervised and supervised machine learning algorithms, and even any statistical analysis methods also, that we will focus mainly and in detail in handling data and how to pre-process and how to work around this problem to understand patterns in the omics data types. And yeah, any other queries so far?
And Daktiva is asking, share the R codes here, please.
Yes, as I showed you in the associated resource, you will get all the codes when you sign up for the program present in the lesson themselves. In terms of Python, again the same, but in Python you will also get access to specific Jupyter notebook or collaboratory notebook that we have prepared for the analysis methods. That this collaboratory notebook can also contain some of additional assignments and challenges that you are encouraged to attempt at, right? All these R codes that we have been that I showed you in the lessons that you will get free access to, and you can copy and paste. Not of course you can copy and paste in your own analysis environment and apply that. If you want specific analysis methods to be discussed in detail, then we have right throughout the program, we have live Q&A sessions at scheduled at different time, and please do attend those Q&A sessions. These live Q&A sessions right throughout this program are for discussing what you have gone through, and if you have any specific queries regarding one specific analysis type that we have not covered in detail or that is not a popular choice, then we can focus on those analysis methods and analysis queries in detail, right? For example, we mainly focus on all the analysis types that are successfully applied to bioinformatics data and biological data and omics data type in general. But we can also compare different analysis methods in this application also. So you are encouraged to do that. And for example, if you need templates for specific R codes, then we might have that in other parts of our lessons that I can point you where to get from, right? And any other queries? I do not belong to bioinformatics or IT. Can you tell once again what this R code is? Antisha, R is a programming language just like Python, and whose environment you will exploit to analyze this data. So we spoke about, and I gave you examples of structured data and showed you loaded that data into Excel by highlighting what happens or what are the entries in the rows and what are the entries in the column, right? And this visualization of this data can be performed, and basic graphing can be plotted in tools like Excel. But it stops there. In order to understand the complex patterns of this structured data, you have to take this data and apply some sophisticated statistical analysis methods in an environment that is suitable to do that. So R programming language and Python programming language is a couple of such environments, right? You can load this file as a table or as an object into these programming environment and apply variety of different methods and statistical analysis that we have been discussing in order to understand the pattern of the data and relate that to the biology of the data that it has been recorded with. So that is what R and Python in a nutshell and how they are involved in these processes, right? If you want more information, you will have more information in our section, in our lessons also, where we have dedicated courses on R and Python as a part of introduction to R and introductory courses to Python, where we start from the basic level of setting up R or installing R in your computer, setting up and installing Python in your computer, and what are the basic commands that you will need to get familiarized with to understand the data types that is related to the biological data and bioinformatics data, and by slowly building up the complexity to push yourself to this level where you are free where your familiarity increases to the level of pre-processing the omics data type in a necessary course to a necessary level where it can be applied to a variety of different methods. So what do we have? Another question. Is there a certificate of attendance? I think Srigauri can answer that. What are the limitations of using statistical tools like SPSS, Innovation, and MATLAB? There are no limitations of using statistical tools like SPSS, but the innate graphical user interface that you will use with SPSS, right? And I have no idea with in vivo stat. And in order to make it user friendly and in order to confine the statistical methods into a GUI that you use for SPSS, for example, SPSS comes with certain limitations and not a full liberty in terms of what you can use and how you can use it, right? For example, I am not very sure about programming in SPSS to apply the same data analysis methods over and over again in a loop environment or in a loop for multiple variety of different comparative methods, right? That might not be possible in SPSS or might be possible in SPSS, but SPSS is basically or is confined to a GUI. Any programming method that is confined to a GUI comes with their own set of challenges that needs to be addressed. And MATLAB, I don't think we have any problem with MATLAB, but MATLAB is not free. That is why we do not focus on MATLAB's version or use of MATLAB for these methods and analysis. Anything that you can do with R and Python, or most of the things that you can do with R and Python, you can do with MATLAB. But as I mentioned, MATLAB is not free. Okay, I think that will answer most of the questions. And over to you, Srigauri.
Yes, sir. So we have like few more queries for Sonalika. That is, do we get recordings of the session? And also, will there be a participation certificate?
Yes, there will be the participation certificates, and also we are going to share the recording with all the participants who have joined today's session via an email. So that would answer your question. And yes. Are there any other questions for me, Srigauri, or can I take up the registration part now?
Yes, we have several queries regarding the registration, scholarship, as well as for the entry requirements. So over to you, Sonalika.
Yeah, so I will just go through that now. Thank you so much, Srigauri, and thank you so much, Dr. Raghav. Now, I would like to take up the registration part. How do you get started with this program? How do you enroll yourself? And what are the available scholarships? So here is the program page. I've shared it in the chat box as well. Once you visit this program page, we have put together everything, including the schedule and also the registration link. So here is the session schedule, where you can see on which all dates the sessions are scheduled to be hosted live. And then you have here the details of the topics that will be covered in each one of these sessions and the associated resources that you need to cover as a part of the session. So as Dr. Raghav mentioned, that there are several resources that you leverage from as a part of this program. So you have to go through these courseworks on the OmicsLogic Learn portal. And as a part of each one of these sessions, you are supposed to complete some of these lessons, including the quizzes and assignments as well. So we have listed them out here. And once the session is complete, the recording will be uploaded on the program page. It will also be emailed to all the participants, but it will also be there on the program page. So you need to complete the associated resources, revisit the recording or watch the recording, and click on a complete session button that will pop up once the session is completed. And doing so will help you get a certificate on your profile tab. Now, these are the list of all the sessions that you can read through as a part of this program and get the information on what are we going to cover for the program. The program starts on September 28th. That is, we'll be having our first session then at the same time. Now, if you scroll down, you'll get 3 levels, which you can pick and register for. We have a subscription level for 45 days. Then you can either subscribe for 60 days or 90 days. So these different days of subscription types comes with different accessibility to the resources that we have on the OmicsLogic Learn portal, the T-BioInfo server, and also the mentorship. So if you take up the 45 days of the enrollment duration for your program, then you will be a part of the training program where you will be a part of these live sessions, go through the asynchronous coursework, watch the recordings. And then if you want to apply your knowledge on a research problem in hand, then you can maybe join the 60 days of the subscription level where you will get to work on a research question with the mentor and plan your research proposal to get started with your research project. And in the 90 days of duration, you'll be able to complete your training and then also work on a research project and publish it successfully if you get the required number of hours in a day for the program. So that is about the 3 types of durations that you can pick while registering yourself. Either 45 days, 60 days, or 90 days. Now you simply click on this button and proceed ahead with your registration. Now there are several scholarship options available for the interested participants. I'll quickly share the scholarship form in the chat box. So those who are interested for scholarship, please fill out this form. Our team will review your application and get back to you with the possible scholarships available. Meanwhile, I'm sharing this form. You can put your questions in the chat so that we can assist you. All right, here is the form. So this was one thing. And then as a part of this program, you have certain courseworks to complete. Python course 1 and Python course 2, and also R course 1 and R course 2. All right, so we have the courses listed out here as well. And on completion of each one of these sessions, you will get the certificate. Where do the certificates come? Before I jump there, I would like to tell you that if you are not from a US, if you're not from US and you belong to a different country, you can simply click on this currency icon here in the main menu bar on top and pick your currency. And if your currency is not available here, then maybe you can use the conversion tool and look into it. Still, if you are facing any issues, please feel free to reach out to us. We'll be there to assist you anytime. I'll be sharing the email ID of our marketing manager. You can mail her at marketing@omicslogic.com and she'll be there to help you out with your enrollment. So this is the profile section that Sri Gauri went through in the beginning of this particular webinar. So if you visit your profile section and click on the certificates tab, here is where you get all the certificates. So you will be getting one certificate for the program completion. And also for each coursework that you complete, you are going to get a separate certificate. And this profile is a portrayal of your achievements that you can share anywhere on the social media accounts like LinkedIn, maybe to showcase your achievements to recruiters or supervisors where you wish to enroll for maybe a PhD program or higher studies or get a job. That was all that I wanted to mention. Are there any questions?
There is one query. How much is the research training on cancer, that is transcriptomic for chronic disease? Is there any available scholarship for low income? I guess the scholarship is answered. So yes, the research training for cancer.
Yeah, so for transcriptomics for chronic diseases program, I think you might have went through this particular program here on the program page. So yes, if you click on the programs tab, you'll get all the program pages listed out here. This is one of those programs which is ongoing right now. And yes, you can enroll in it. And same goes for this one as well, that you scroll down this session. This is the way you can look at the session recording and complete the session. This is a program which will be concluded soon. So we'll be restarting it. And I am a part of this program page. If you click on the link of this program, you can look the registration costs and pick the level that works the best for you.
Thank you so
So
much,
once
Snehal.
a person is a participant, thank you, Srivari. Once a person has joined the program, then this is what they see. Like they can watch the recording, complete the session, and this is how the recording comes up. And also you get to click on the Zoom button to join the Zoom meeting directly from the program page itself if the session is pending. Yeah, so that was it. Over to you, Srivari.
There is one more query, that is, please share information about research fellowship. Will that involve training?
Yes, definitely. A research fellowship involves training. You can't straight away work with your research question in hand. If you don't have the required skill set, if you don't know about the concepts involved or the algorithms involved, or if you don't have a hands-on with the pipeline. But if you have a good hands-on practice and if you have completed the initial training with us already, then you can straight away start with the research fellowship program. So the research fellowship program is for either 3 months or 6 months. The outcomes of both of these durations are different. Obviously, in 6 months, you are giving in more time to your research fellowship, and the outcome could definitely be as publication. And the 3 months program, you'll be able to work on a poster presentation maybe that you can present in any of the conferences and also competitions. All right, now what is the structure basically? So in the first month you go through the training process, you meet the mentor twice every week, take your doubts from the mentor and proceed ahead. And then when you have to work on your research project, once your training is completed, all doubts are clarified, and you have a good hands-on practice with the pipelines as well on the T-BioInfo server. After that, you're supposed to submit a research proposal to us. So you review literature and come up with a question that you would like to work on. If you don't have a question with you that you would like to work on, but you have an idea what exactly you want to work, then mentor will help you just design your question, design a framework for you, and then get you started with your research project, sorry. Now, once you get started with your research project, it remains the same that you will be meeting the mentor twice every week to update the mentor with your progress, to clarify a question that you might have, and discuss the next steps. And these meetings are one way that we communicate, and we have Skype groups set up for each research fellow. So you are in touch with the mentor 24/7 on weekdays specifically, where you can ask the mentor your question or mention the step where you are stuck, and we'll be there to help you out. And if there is a meeting required in between, apart from the 2 live meetings, then you can ask us and we'll be there to help you out with that. This is just a glimpse of what I've shared. There's a lot that happens in a research fellowship meeting, and the program definitely. So we'll be shortly organizing a research fellowship webinar as well, where we'll discuss about different specialization tracks as well on which we are working. So there are 6 different specialization tracks on which we are working. I'll just take few minutes and give you a brief overview. So one of them is oncology. Then we also work on precision medicine, infectious diseases, space, omics, agriculture, biotechnology, and neurosciences is one of them as well. So these are some of the fields that we are working on and applying, obviously, omics data analysis skill sets. So that is what we are working on. This is just a very brief overview of the specialization tracks, and we'll be discussing more about it in detail in the webinar. So if you are interested in the research fellowship program, what you can do is just open up this program page and maybe submit your interest. If you click over here, register program interest, just submit your interest. And whenever we have this webinar, you just receive an invitation. You can join and then take up your questions. Metagenomic analysis on your water samples working to determine the algal community structure of some water bodies. So when you say your samples, did you collect these samples and get them sequenced? And do you have your own data that you want to work with? You can unmute yourself, I think so. Yes, you can. Yeah, please go ahead. I can't hear you. Okpanachi, I'm not sure if I'm pronouncing your name right either. Yes, I can hear you now. Thank you.
Yes, I said I'm planning to collect the water samples. I cannot just collect the water samples without sequencing immediately and storing it appropriately. So I'm planning to collect the water samples. So, and I actually want to get informed knowledge about bioinformatics.
And do you come from a bioinformatics background, and do you already have an idea about what pipelines you could run and which all are the algorithms involved for the analysis? Do you already have an idea or not?
No, I don't. It's actually part of my thesis. So, and I just had to do it's part of what I meant to do in order to complete my PhD work. So, and I want to learn as I'm here.
Got it. So for you, I would recommend the metagenomics program maybe. So here is the metagenomics program. And in this program, we will first of all get you trained and help you understand what is the metagenomics data all about, all right? And then we'll discuss about various pipelines that you can use. There are two very popular pipelines for metagenomic data analysis. One is Data2 and the another one is KIND2, which helps you look at the microbial abundances of your microbial communities in these samples, and then also calculate alpha and beta diversity, and also look at the taxonomic abundance. That's what I mentioned. And then also you calculate the core matrix, all right? So the distances for each one of these samples. All right, so there's a lot that you cover in metagenomics. So here is the program page for the metagenomic data analysis program. The program already commenced yesterday. We had the first session. Please drop your email address in the chat box. We'll share the recording of the first session with you. And you can follow along the session schedule here to understand what all topics are we going to cover and how this program is scheduled, to give you an understanding. And talking about the research aspect, you said you're going to have your own samples. All right. So when you have your own samples, we can facilitate an FTP account setting for you where you can upload your samples and then use the SVL links of those samples location to run these pipelines on the T-BioInfo server and help you with your analysis. All right. So I will suggest that you pick the 90-day subscription level because you want a full-fledged research project that you want to work on after the training process is over. And we are also providing scholarship for this program too. So the same form you can fill for this program as well. All right, you just need to pick the program. Yeah, just fill this form and drop in your email address so that we can share with you the details and we can help you with a good scholarship to get started.
Thank you.
That's it. Welcome.
We have one more question. Are there any charges for metagenomic hackathon registration?
Yes, there are charges involved for metagenomics hackathon registration. And we are going to launch the program next week, and we'll be discussing about the charges in detail then as well. But I'll just give you an idea about it. So if an individual is registering, it's $100, and if a group of 3 people are registering for the hackathon, then it would be $300. And these group of 3 people should be like, one should be a researcher, and another one should be from a computer science background, and the third one should have a business background. All right, or maybe understanding of how to apply the project that you work on to a business setting. And if you don't have the 2 partners or one of them that you could make a group with and register, maybe you can register individually, and then we can help you get into a group and then start working with your analysis, and then finally apply it to the industrial aspects. Yes, that's pretty much about hackathon. And Ola is asking, do you help students in building their own tools? So that will again be a part of the research fellowship program, Ola. And Dr. Raghav, would you like to add on to Ola's question?
Sorry, what's the question? Do you help students in building their own tools?
Building their own tools, yes.
If you are going to sign up for a research fellowship program, and then depending upon what you can understand, what you can achieve within that time frame for building up these tools. So we, to address on the possibility of such a project, we have to get up, or we have to set up a separate meeting with you, Ola. So it depends on the complexity of the question. It depends on what you want to write, or what you want to include in these tools, and what are the analysis steps that you want to include, and what you want to analyze. So if you don't want to be specific in this public forum, you can get in touch with us using support@pine.bio, or you can you can also write to support@pine.bio. If you're free to set up a meeting with us, we can go about by understanding more specific information about what you want to do and how feasible it is within the framework of research fellowship program. Okay. And there is another raised hand by Adamu. Yes, Adamu.
Good evening. Can you hear me?
Yes, I can hear you. Yes, please go ahead.
I am Adamu Muhammad Ibrahim. I'm from Nigeria.
Yeah.
I'm a medical laboratory scientist. I have a passion on bioinformatics, especially infectious bioinformatics. So I am asking, what are the basic requirements for someone that wanted to start a career in a bioinformatics with a molecular biology background?
Okay, if you want to start up a career in bioinformatics background, you have to find ways to expand your knowledge in some sort of coding. I don't have to learn many things, but learning one coding language will help you associate with the others also. So bioinformatics skills mainly involves using different analysis methods and then mending these analysis methods to suit your data and the question that you are studying. So for this, you can start with understanding the logic behind this analysis method in several of the resources that we are sharing with you. And if you have an environment like T-BioInfo Platform, and within what this platform offers you, you can start with code-free analysis of bioinformatics data analysis. But eventually, if you want to be a separate bioinformatician, you have to get yourself familiarized. Yeah, you have to get yourself familiarized in one coding language, preferably R or Python, depending upon your nature and your comfort level. Okay.
Okay, thank you very much.
Cheers.
Thank you so much, everyone. So with that, we'd like to conclude today's webinar. Thank you, everyone, for your patience and time. And I hope to see you all for session 1 of the data science program. And if you have any further queries, I've shared the details of the marketing manager. You can reach out to her via her email ID or her phone number, which is also active on WhatsApp. So thank you, everyone. Have a great day.